Voice of the Sea, learning from experts across the ocean.
Welcome to Voice of the Sea.
In this episode, we're learning about the technology that researchers at the Hawaii Institute of Marine Biology use to study genetic relationships, conduct long-term experiments, and remotely assess the health of coral reefs. The location of the Institute within Kaneohe Bay is key to understanding our island populations of coral, fishes, crabs, sponges, and other organisms. Lead researcher Dr. Rob Tonin describes why.
HIMB, Hoku'oloi Coconut Island, is unique. There's nowhere else on the planet that you have this modern molecular facility with the ability to do the types of work that we do right here beside a live coral reef. We can actually work with live coral right here, and we can do those experiments, not have this baseline stress that's associated with transporting things that you have to slosh them back and forth in a bucket for half a day. Kaneohe Bay is a really unusual place, not just in Hawaii, but in the world. We have an enclosed bay with a barrier reef that sort of limits the amount of water flow. Because it's this big, very shallow bay, it warms up. The water stays there for a long time, and so it's actually naturally warmer than the surrounding oceanic water. And there's so much life in Kaneohe Bay, and all of those things are breathing. Everything that breathes puts out carbon dioxide, and so it's actually also naturally acidic. And so Kaneohe Bay is this fantastic giant aquarium for future climate change research because it actually is an ocean of the future already. And the corals in Kaneohe Bay are actually these super corals out of the entire population that are able to withstand the high temperature and low pH. If you're familiar with the history of this area, all of the corals here are new too. We used to have the sewage outfall for the city of Kaneohe in the south part of the bay. Dumping raw sewage in here made the whole bay pea soup green. Not a great place for corals, and we lost most of the corals in Kaneohe Bay. And so when they fixed that in the 70s and moved the sewage outfall offshore, Kaneohe Bay bounced back, and almost all of the corals here have come back since 1976. Over the course of a year, what we see so far is the corals that went into the tank have the same tolerances, the same ability to handle temperature and pH stress going into the experiment as they have after a year. And it looks like it's driven by the genetics and not by the exercise or the physiology of the organism.
How is looking at a coral's DNA or an opihi's DNA going to help you manage it for future generations?
If we think about like the opihi example, and if we imagine that you're looking at a family, if I look at the DNA and your mother, your grandmother, your great-grandmother, all the way back are on the same island as you are, there's a very good chance that your offspring and the next generation are also going to be on that same island. But if I look at it and your grandmother is from the Big Island and your great-grandmother is from Kansas, then there's almost no chance of us being able to predict where your offspring are going to go either. They're moving around. If opihi are moving back and forth between the islands, we need to think about the population as a whole across all of the main islands. If, as we find, opihi almost never cross the channels, every island is by itself, it really doesn't matter how many opihi are in Maui because that's not helping Oahu. And what we have here is where our next generation is coming from, and what we do to our own backyard is what will determine what our kids are going to see.
The main question that I'm trying to investigate from the genetic work is how connected are the different crab fishery populations across the islands.
Fill me in on our crabs.
Yeah, so we have a number of crabs. We have over 200 different species total. Yeah, not all of them edible. In sort of traditional culture, we had actually quite a few different crabs that were consumed. The research I'm doing involves the Samoan crab, and then also a native crab. That one we call kuhonu or white crab or haole crab or white crab poke or kimchi poke. Kuhonu is subtitle, so you'd find find it. You could find it on sand flats. One of the things that makes it really distinct are the 3 red spots on the back. They can get up to a few inches. The scientific name for kuhonu is Portunus sanguinolentis. Sanguinolentis referring to the fact that it comes from Hawaii. This one is Samoan crab, so this is the introduced one. Remember how I was saying it's a monster? Yeah, because look at how huge it is. In Hawaii, we call it Samoan crab, but we call it that because they were imported from Samoa. It has spread across the islands since the 20s. It's found pretty much on all of the main islands. There's some articles from the 20s, even in the newspapers, that were saying, like, you know, with this crab, like, it's such a problem, we need to lift regulations and we need to go after it because it's even doing things like burrowing under fish pond walls and destabilizing. Okay, so we know when it was brought in, we know about how many were brought in, and we know where they were brought into. But we don't know anything about seasonality in the islands, right? We don't know when they're reproductive. We don't know how often. We don't know the maximum size that they reach in the islands. We don't even know where the gravid females are, the females that carry the eggs. We have no idea where they are. We don't know where the larvae land. So there's actually so much about them that we don't actually know. Samoan crab, you could actually find it definitely subtidal, so definitely underwater where there's mud and where there's mud sand mixture, where the mangroves are. But in some places, you can actually find them a mile plus upstream. They can end up really far inland, which none of the other crabs that we have, at least currently, do that. For the 2 crabs that I study, this is where they live. They don't live in the northwestern Hawaiian Islands. By collecting this tissue and then using it to compare it across the islands, one of the things that we can try to understand is a fish pond or is a area on one island self-seeding, or is it very well connected to everything else? And then for Oahu Island, I'm also collecting along the coastline so that we can dive a little bit deeper and sort of ask the question, are these crab populations connected? How well connected are they along a single coastline? If you were a tiny little baby floating around in the plankton, you might be able to start off at Hawaii Island and make it all the way up to Kauai, which we don't really know from physical evidence. We don't know from looking at the organism, but we can tell that from looking at its DNA and making comparisons across the islands. You would want to know if you were on one part of any island, where are your stock coming from? Are you self-seeding in that, you know, you're going to control your population in a totally different way than you would if your crabs were coming from all over instead, right? Then you might be a little bit different. You might want to coordinate with your neighbors or whoever the crab are coming from so that you guys can both work together. If they have, you know, the source population, you have the sinking population.
The University of Hawaii's Sea Grant College program, focused on Hawaii's coasts and its communities through sustainable development, safe seafood supply, sustainable coastal tourism, hazard resilience, and healthy coastal ecosystems. Hawaii's Sea Grant.
So tell me what we're going to be checking out here.
So we've got a big array of mesocosms over here with corals and other critters growing in them. A main focus of this project is looking at the potential for corals to adapt to higher temperature, higher CO2. So we're looking at several different species, all of our major reef builders here in Hawaii, collected from several different locations around Oahu. Knowing who's going to make it and who's not is critical to being able to have reefs and have fish and have storm protection and have tourism that we get from reefs. And here we've got all of our dominant reef building coral species. We've got 8 different species of corals in here. They're by far the most important contributors to what's generating our coral reefs around here. You can see in some of the tanks, we've got lots and lots of happy, healthy, rapidly growing corals. And in other tanks, growing under higher temperature and higher CO2, like they will be in the future ocean, some of the corals are doing really well. Others are not doing well at all. Others have already died. A big question that we're looking at is what is the potential for these coral species to adapt to those hotter, more acidic conditions that they're going to experience in the future. And the good news that we're finding is while we know some of these corals certainly are going to do poorly, as you can see, some of them have died, others are doing very well. And that gives us some hope that we can actually transition to a future where those more tolerant individuals start growing and growing and growing more on the reef. And even though things will be warmer and more acidic, we can perhaps have a coral reef that's still healthy and built by those sorts of individuals. A few things have been quite surprising and quite cool. One of my favorite corals that we have here is blue rice coral, Montipora flabellata. Oftentimes, if you go to real wave-exposed areas, it'll be glowing bright, bright blue. It's a gorgeous coral. In prior coral bleaching events, we've seen that coral is often very, very sensitive to high temperature stress. And sure enough, we found some of the corals of that species that we collected are very sensitive to high temperature stress. But on the flip side, a lot of them are very, very tolerant. Something that we did not expect at all, no one would have expected, is that we're seeing a lot more babies of one of our common corals here, lace coral, under future ocean conditions, under high temperature, high CO2, particularly under high CO2. That shows the value of doing a long-term project like this one. This is a 2-year experiment. We never would have seen that if we ran a 1-month or 2-month experiment, but over the course of, it's been going about 1.5 years now, the pattern is emerging really strongly. Our corals literally are growing twice as fast as they have in most previous experiments using tank systems like this. So we think we're getting a lot closer to the natural system. All the corals are happy and healthy and look great, right?
And each mesocosm here appears to have a fish.
Yep. So we've got 2 species of fish. They get rotated through. We've got a butterfly fish, which eats natural coral parasites. So things like Vestella, which come in and eat these parietes, things like that. They are eating coral parasites, just like they do out on the reef. If we didn't have the fish in here, half of our corals would be dead in no time. So we've got natural sunlight attenuated with shade cloth so that we've got light levels that are similar to 5, 10, maybe 15 feet deep where these corals were collected. We have a lot of water flow in these tanks generated by seawater pumps as compared to what is often used in these sorts of experiments. If you go to a coral reef, pretty much anywhere, even a relatively protected one, you've got waves. You've got a lot of water motion, and that water motion is important in driving all sorts of metabolic processes in the corals and the other organisms. So we have 2 levels of temperature, 2 levels of pH. If you cross those 2 factors in the 2 ways, you end up with 4 treatments. So low temperature, normal pH is the control. Low temperature, low pH is the ocean acidification scenario. High temperature but normal pH would be just the climate change scenario, and then high temperature plus low pH is the more realistic future scenario. Is the high temperature killing the corals? Is the high CO2 killing the corals? Is it a combination of the 2? By splitting the factors up, then we can tease apart which factors are causing which responses, and when you have them in combination, does that relationship change? The fact that we're seeing corals that can not only survive, but do quite well under these warmer and more acidic conditions that they're probably going to be seeing in the future, gives me a level of hope for our coral reefs. But I also know that those more tolerant individuals only have a chance if we solve our local problems facing coral reefs. Problems of sediment washing onto the reef, problems of pollution, problems of overfishing. They may be able to tolerate higher temperature and higher CO2, but that doesn't protect them from seaweed. That doesn't protect them from mud. That doesn't protect them from the things that we can do on land that either kill them or keep them alive.
Should I just start talking about arms?
Sure, what is this?
Okay, so this is an arm, so not literally your arm, but it's an autonomous reef monitoring structure. And this is a really great way for us to look at the reef without taking a part of the reef and bringing it back to our laboratory. So what this is a way for us to assess biodiversity. And the way that we do this is we put this out in the field. Here, we put these inside of these tanks that have different levels of acidification and heating to simulate climate change. And this gives us an idea of what we expect the biodiversity or the recruitment to look like in the future. This one is a mini arm, so it has 3 different plates. We've had these in there for almost a year, and every month we take them out and we photograph them. Oh. And we're trying to get an idea of what's growing on them, and those things that are growing on them, how are they interacting? And this gives us a better idea of, okay, under ocean warming, what kind of organisms will we see in terms of sponges, tunicates, crustless coralline algae, other things besides corals. And the really cool thing that we found, it's really cool, we've seen a lot of sponges. These are cryptic sponges that have not been described here in Hawaii. So they're new species of sponges. There's about 6 to 8 different species of sponges that we found that have not been found or described before. These are new species of sponges. And we're also seeing that a lot of the sponges that are recruiting under our ocean acidification scenario here are calcareous sponges. And we wouldn't expect them to grow under ocean acidification scenarios. So that's really weird. So what is going on there? I don't know, and we don't know, and we're trying to figure it out. We're seeing different organisms are recruiting on different parts of the arms, and they're interacting with each other in interesting ways. When this project is done in about another 6 months, we'll have an idea about what the biodiversity looks like under these different levels of ocean warming, ocean acidification, and the combination of both of those together. And that will give us an idea of how the reefs here in Hawaii will look like. And this is really important for managers here, so they understand how the reefs will be changing. Recently, what we've been seeing is as the water temperature is getting warmer, the corals are starting to bleach and they're losing their color. You have the coral animal, and inside the coral, you have a dinoflagellate or a microscopic algae that lives inside. And that algae provides about 90% of the diet for the coral. And when the water gets too hot, the coral kicks out the algae, or the algae leaves. All of the algae is being expelled from the coral, and the coral becomes a light color, or it becomes white. Mm-hmm. So when you see the color of a coral, color is the algae. We've had 3 bleaching events here in the main Hawaiian Islands, one in '96, one in 2014, and one in 2015. What we've been trying to do is look at the color of a coral and try to get an idea of the health of the coral based on the color. If the coral is stressed, it will change its color. It will get lighter in color because those algae are being expelled. So can we correlate that color with the health of the coral? To develop this card, what we've done is we've taken 6 dominant species here in Hawaii that have a variety of different colors, and we've simulated a natural bleaching event. So we've increased the water temperature, and we sample those corals every so often to get different colors of that species, to get an idea of, okay, this coral is this color. That means it has this amount of algae that's performing at this type of level in terms of photosynthesis.
Do you need to know what species of coral you're looking at to use the card?
No, you actually don't. What we would like to do is we'd like to combine this color card with what's already being done. It's a grassroots movement. That's the Eyes of the Reef. Yeah. And what they're doing is it's a community-based reporting system where you're like, oh, there's bleaching going on here, and you report it. And we can get an idea of how healthy that coral is in that area. And we've also been working with the Division of Aquatic Resources to implement this into the coral bleaching response plan. So when there is a coral bleaching event, the scientists, managers, community members can go out with this card and assess how bad the bleaching is and what types of species of corals are bleaching and how fast that bleaching is occurring.
So they're all using the same type of ruler.
Exactly, exactly. And this is going to give us an idea of not just what's happening here on this reef right here, but what's happening in Kauai, what's happening in Maui, Big Island, all across, the Hawaiian Islands.
The Curriculum Research and Development Group in the College of Education at the University of Hawaii at Manoa. CRDG's training roots go back over 40 years. Through professional development programs, curriculum workshops, research on teaching methodology, individualized school and district training, and so much more. The Curriculum Research and Development Group, improving schools, improving education. CRDG.
Next, we're taking a look at the colors reflected by healthy coral versus coral rubble and other substrates with Dr. Dan Shar, who is working with NASA researchers to remotely assess the health of coral reefs.
Reflectance is just the light that comes off of something. The sunlight's hitting this flume. You see this as green, you see this as white, you see something in the bottom is brown. So the light that reflects off of something has different wavelengths. That's the color we see. So what you can see in a coral, if it has healthy pigments, you'll see one set of spectra coming off of it. And if there's something that maybe is dyed, it's whiter, so you'll see a different spectra come off. We can start to use that information to quantify the health of coral in remote locations. A flume is a device that it's using water that's drawn in from out in Kaneohe Bay, and it has a motor, and it's just recirculating water at a set speed. I have this water moving between 12 and 15 centimeters a second, and I'm able to completely shut it off. So if I had coral in here, I can measure the coral's respiration, what oxygen it's using, what CO2 it's putting out, and I can also measure its growth, its calcification, because it's a closed off system. So by flume, a flume is just, I think, a channel of moving water. But this one is what's called a racetrack, where it moves this way and it circles back that, what goes under here. And I just use a small boat trolling motor to regulate it. And I have an instrument down there that's acoustic Doppler sensor that gives me the exact flow speed. This is a smaller part of a larger project that's funded by NASA, where they have an instrument called PRISM, which is a large instrument that's mounted on the bottom of a plane, and they're making measurements of the light reflecting off. And I'm building a database that will allow us to look at those larger measurements and say specifically what things are, such as coral rubble, or very healthy coral, or dying coral. The instrument they use is, as I said earlier, it's called PRISM. It's like one of these instruments, but it's large, and it's maintained in a vacuum. And so they have to keep it cold in the belly of this airplane. And it goes up and it flies at 29,000 feet, and it will stay up for a couple hours and fly around. But it has to be in a very controlled environment and very stable. We had a plane here for a month, and it was able to scan every coastal region of all the Hawaiian Islands. And then it was in Guam, and then in Palau. And they flew around any islands around those areas. They went into, the project went into the Great Barrier Reef and did Lizard and Heron Island, and it's going to do Bermuda and coastal Florida as well. The idea is once they perfect it on an airplane, then can move to satellites. As resolution is able to increase, you can actually have a satellite fly over and scan a large area. But that's a whole other project. One of the benefits of doing projects like this is it allows people to start to get almost an early warning sign to the health of different, like reefs and different environments, without having people actually have to go there. It's really difficult to have people go every little spot and constantly check stuff. But if you could just have a plane or something else that flies over an environment, they can start to say signals like, this reef doesn't look that healthy, something might be happening there, we should go look at it. All the data we collect is going to be freely available for other people to come in and interpret. So it's more like the project is funding the instruments, the vehicles to make all of this measurement, and then other scientists can then start to download it off the NASA's website or the Jet Propulsion Lab site, and actually they can start to make answers on what some of this stuff is.
Exploring Our Fluid Earth is a dynamic curriculum developed by the University of Hawaii's Curriculum Research and Development Group, teaching ocean science concepts through the disciplines of physics, chemistry, biology, and ecology. Exploring Our Fluid Earth is now available freely online. Find out more at exploringourfluidearth.org.
We're looking for a few heroes, mentors, trailblazers, innovators, a passion to change lives, spark curiosity, open hearts, and awaken minds, help students answer the question, who am I? This could be your calling, but this is no job. It's the journey of a lifetime. Be a hero. Be a teacher.